Okay, folks, we were talking about metabolism, which is the sum total of all reactions in a living thing. And this would include anabolism, where we build things up, we put energy in to make bonds, and catabolism, where we break things down. And energy would be released by breaking bonds. Now, if we wanted to make bonds in a chemistry lab, we could do this by just simply adding energy in the form of heat. We could turn Bunsen burners on, put the reactants in there, and they might form bonds. Well, in the case of a living thing, a lot of times there's a certain temperature that we cannot exceed. Okay, so look at a human body, for example. We have a certain stable temperature, and we can't just add heat to make reactions go. What we have are enzymes, of course. These are molecules that catalyze these metabolic pathways. They help with the making and breaking of these bonds. And what they do is they decrease the activation energy. They decrease the energy necessary to make that reaction go. Here in this picture here, we've got a certain amount of activation energy that is going to be necessary to make this reaction go with the enzyme, and we have a lot more energy we would need if the enzyme wasn't there. So it decreases activation energy, and these enzymes, as a result, increase reaction rate. They make the reactions go faster. Otherwise, we need to add heat, okay? Now, we will need to know a basic mechanism of enzyme activity, all right? So here we have an enzyme. Enzymes are typically proteins, not always, but let's just say this one is a protein, and proteins have a certain three-dimensional shape because of those hydrogen bonds we learned about way back in the chemistry review. Now, here in this case, the shape of this particular enzyme gives us an active site that will fit, in this case, two substrates, and they happen to be amino acids. All right? So they get close to one another, and as a result, there's enough energy here now to form these bonds. This happens to be a peptide bond between two amino acids. So in this case, it happens to be an enzyme, the enzyme which is a protein, and it is taking part in building up another protein in this particular example. So we have three basic steps. We have the substrates that bind active site. We have certain internal rearrangements, whether it's a making or breaking of bonds. In this case, there is a bond made. And then the product was released. Now, it's key to understand that these enzymes are reusable. They can be reused again and again. And a lot of enzymes will also help to catalyze reactions going the other direction. So in this case, imagine you wanted to break that peptide bond. Now, enzymes are like this in all living things, and we need to have this basic idea down, all right? So here's another picture showing the same basic kind of thing. We've got an enzyme. In this case, if we go this direction, it looks like this particular enzyme is taking part in breaking the substrate rather than making a bigger product, right? It's going to have two smaller products. But still, it's an enzyme. It's got a certain active site. Same basic idea. Now, to totally understand how an enzyme works, we have to understand that some enzymes need cofactors to function. They cannot work on their own, okay? So here we have a basic idea of what an enzyme can do. Here we have an example of an enzyme that needs a little help to actually catalyze a reaction, right? So here we have something called an apoenzyme. This is a protein just like the earlier enzyme we were looking at. But in this case, the active site will actually not accommodate the substrate. You can see the substrate over here, right? This substrate over here has a certain shape that it needs in order to fit. Well, this is not that shape over here on the left. What it needs is the binding of something called a cofactor in order to form the active site. Now, cofactors can come in the form of minerals or ions, like, here we're talking about electrolytes like calcium or magnesium or iron. Sometimes cofactors come in the form of organic molecules, and then what we'd call them coenzymes, like vitamins. Vitamins are oftentimes cofactors for enzymes. So here, once that cofactor binds the apoenzyme, we have just the right shape, and now the substrate can actually fit in that active site and catalyze that reaction. All right? Now, we have to understand that enzymes like these are in some living things and not in others. And a lot of these living things are totally dependent on these enzymes in order to live, right? So to give you an idea of how our knowledge of cofactors has given us the ability to adjust and modulate whether bacteria grow, one good example would be fluoride, right? It so happens that fluoride has the ability to bind certain cofactors like calcium and magnesium. Now, bacteria in our mouths are dependent on certain types of enzymes that need those cofactors. So, if they put fluoride in your toothpaste or some in the water or something like that, the idea is that that would actually bind up all of those cofactors. And if it binds up enough of those calcium and magnesium cofactors, the enzymes that these bacteria are dependent upon will not work. Now they've learned that fluoride also does other things. Fluoride binds some of these cofactors, which early on they thought that was the only thing it did, and it would help to stop bacteria from causing tooth decay, right? But they've also learned that fluoride actually does something to the surface of our teeth, and it makes it harder for bacteria to actually attach to our teeth as well. So there's really more to it than just that. But this is an example where hinging upon the cofactors and the bacterial dependence on these enzymes allowed us the ability to actually change things a little bit there in that environment. Now, we also want to know some of the factors that influence enzyme activity. One of the factors would be temperature, right? In general, as we start to add heat to a situation, enzymes move faster. The substrates move faster, the enzymes move faster, all molecules do. And as a result, as we add heat, there will be more instances and opportunities where the active site of enzymes will come in contact with their substrates. So more enzyme movement, more molecule movement would result in higher enzyme activities. So here we see temperature going up, and as the temperature goes up, we see enzyme activity goes up. Now there's a point where it's really good, and then it really drops off. Well, this would be a situation where you had too much of a good thing and denatured those enzymes, right? Remember that hydrogen bonds hold these enzymes together. So at a certain point, yeah, it's really going to increase the rate of reactions, but then if you go too far, those enzymes will actually be damaged and the rate of enzyme activity will decrease. Another factor that influences enzymes is pH. Again, it's a lot to do with the hydrogen bonds that hold these enzymes together. So here we have a pH scale, okay? pH scale going from 0 to 10, okay? Doesn't go all the way up to 14. They're just showing the activity for a certain enzyme here. And in general, you see that as we increase the pH up to around 5 here, it looks like 5 is perfect for this particular enzyme. you're going to have more activity. And this has to be because that particular pH will help to maintain those hydrogen bonds best for that particular protein. Okay? But at a certain point, if this pH goes even up further, we would see a decline, a situation where these proteins would denature. So if we have too much heat, or if we go outside of the range that is good for the pH for that enzyme, we're going to see that it's not going to function, right? Finally, we have the substrate concentration. How much of the reactant, the substrate, that the enzyme is going to act upon makes a difference, okay? So let's look at this graph. This graph looks very different, right? Here, if we think about it now, if there's very little substrate, I would expect very little enzyme activity on those substrates, right? If this is zero, if we have zero substrates, we'd have zero amount of enzyme activity on those substrates. But as we add and add and add substrates, well, of course, the enzyme activity would go up. We'd have more reactions taking place. Now in this graph, though, we reach a certain point where the enzyme activity cannot go any higher. And this would indicate that all of the enzymes are busy. All right, so let's say you have 100 enzymes, and we now put in 100 substrates for those enzymes to work on. Well, that's about as fast as those enzymes can go, right? Only 100 enzymes can be used at a time. So we see a plateau here. We see a limit to enzyme activity based on the amount of substrate that happens to be there, but can only go to a certain point. Okay? All right, one last thing I want to go over with you guys would be enzyme inhibition. How can we stop enzymes from working? One way that we can inhibit enzymes is through competitive inhibition, where a molecule, other than the substrate, competes with the actual substrate. So here in this image, we have an enzyme here in the bottom left, right? And this enzyme has just the right shape for our substrate. Well, nothing would stop that from happening, right? There would be a reaction here as a result of the normal binding of the substrate to its active site. All right, so now let's move over to part B here, okay? Here we have a situation where this yellow molecule has some shape similarity to the natural substrate. And it has found its way into the active site to bind there. But now the substrate itself cannot get to the active site. The yellow molecule is competing with that red substrate. This is called competitive inhibition, okay, where a molecule binds the active site, interfering with the natural binding of the substrate. Okay, an example of where we use this is with sulfa drugs. Okay, let's look at this image here on the top left. This particular molecule in the very top left, we'll just call it PABA, okay? This is the substrate for a certain enzyme that's found in bacteria. Okay, so and bacteria have enzymes that work upon PABA, this substrate, and ultimately as a result, they make nucleotides so that they can actually make DNA and replicate and grow. Okay, so pretty important enzyme, right? If this enzyme uses this particular substrate, PABA, to make nucleotides and DNA, that's really a critical function. Now let's look at this molecule. This is a form of sulfa drug. Looks very similar, does it not? Looks like these molecules are almost identical. In fact, what occurs with a sulfa drug is that the sulfa drug competitively inhibits the natural substrate, PABA. So bacteria can't make their DNA anymore. Now, animals are not dependent on this enzyme. We get our DNA in a slightly different pathway. We don't need the enzyme. We don't have that enzyme. So sulfa drugs do not interfere with our own metabolism in that way. And so, of course, we use sulfa drugs for antibiotics quite a bit. Some of you might be aware, though, that sulfa drugs can cause allergies in some people. So for some people, sulfa drugs are not a good alternative. But for many, they are, and they work as a competitive inhibitor. Okay, they also have what are called non-competitive inhibitors. Okay, these bind someplace other than the active site. So here we have a picture down here in the bottom right. We have our enzyme, and that enzyme started with the shape that we see over here on the left. Okay, it started with a shape that a natural substrate would bind. However, now what we see is a non-competitive inhibitor has bound here in some other place other than the active site. What this has done is it has altered the three-dimensional shape. It's altered some of those hydrogen bonds in there, and now our shape is not going to accommodate our natural substrate. Non-competitive inhibition. Now, give you an example of a couple other drugs that some of which are competitive inhibitors and some of which are non-competitive, and some of them do both. All right, and some of these are arsenic, cyanide, mercury. Lots of heavy metals happen to be inhibitors like this. Some of them are competitive, some of them are non-competitive, but they're deadly, right? These are all poisons to the human body as well as to other living organisms. And so this also tells us that some inhibitors are not specific, right? I wouldn't want to treat a bacterial infection that I have with any of these compounds. They might kill E. coli, they might kill Staphylococcus aureus, but they kill me too, right? So these are happen to not to be as specific for the enzymes that just bacteria use. All right? So we'll leave it at that, guys, and we'll dive into more metabolic pathways next time.